All right, thanks everyone for joining us today. It is my pleasure to introduce Julien Vermot from Imperial College London. His research focuses on understanding the impact of mechanical stresses during morphogenetic and regenerative processes. So today he will share with us some of the related work about mechanical forces and cardiogenesis.
Yes, thank you very much for the introduction and the invitation. Really happy to share with you our work on a topic that we are interested in for many years now, which is trying to address the impact of mechanical forces during the process of cardiogenesis. And in general, this is a topic that is fascinating and is related to many processes in the embryo in general. So it's very well known that we start as a fetus to have an activity, a mechanical activity, before the organ is entirely functional. So you can see here a human fetus after something like 3 months, I think, of growth, and you see that it's moving, it's really active, but obviously the skeleton and the muscle are not developed and properly formed in this individual. And it turns out that the mechanical activity and the process within the cells at these stages are really important when speaking about physical stimulation, are very important for the growth of the bone and the muscles at these stages, as well for the nervous system, which will connect the muscles associated with the bones. In addition to this, so there is another system which we are interested in, which is the cardiovascular system, that also gets active much before it's fully developed. And you can see again a human fetus, and you see that the contrast is very bad, but there are some movements here, and it's due to the activity of the heart within the fetus. It turns out that like for the skeletal system, the musculoskeletal system, the heart function is really important for its proper growth. So it raised the question of mechanical forces. What do they do? And we are amongst people that are really interested by that, trying to understand how mechanical forces affect morphogenesis. Turns out that mechanical forces are everywhere. They are acting at every scales on biological system, from bacteria, plants, and of course mammals. We use those physical information to comprehend our environment, but in the embryo there has to be some elements that sense these mechanical forces and tell the cells to do something, and in particular promote growth and morphogenesis. So our question is very fundamental and very basic, and it's basically to understand how mechanical forces regulate gene expression and cell behavior during the process of morphogenesis. And we focused on a specific type of mechanical forces, and actually we thought it was very specific, but it turned out to be very broad, and we had to turn to many other type of forces. But overall, we are focusing on the forces that are generated within the cardiovascular system, which are associated with flow and red blood cells movement within this network of vessels that is growing. So to do this, we use an animal model, which is a zebrafish, which is an animal of, when it's adult, a couple of centimeters long. But as an embryo, it's very easy to manipulate and very easy to image. It's very resistant to light. And there is a wide array of transgenic line that labels different subtypes of cells, such as red blood cells or the blood vessels themselves. So we can, with high accuracy, explore and quantify the forces generated in the vascular network. And we can also record the response of the cells to these forces in terms of a morphogenetic process. So you can see here the vascular system in a larvae of 3 days. You see it's still immature, so you have some vessels that are made, but you also have some vessels, for example, in the head here, that are still in the process of forming, and there is a strong activity. Now, importantly, throughout the process of the formation of the cardiovascular system, the heart is going to be beating. It's the first organ to be functional in the embryo, and it will generate these flow forces throughout the vascular system. And these forces are really important to coordinate the formation of the entire cardiovascular system. So our question is really straightforward. We try to understand how mechanical forces affect shape, and vice versa, as shape is changing, how forces are affected by that, and how this interact with each other, so that eventually you get a nicely formed and well developed structure. So one of the most exciting area of the body for us is the heart, because it's certainly the structure that is the most mechanically challenged throughout our life. And it does so as a response of a contractile activity of a specific set of cells that form the heart. And these cells are forming the so-called myocardium, which is the intermediate cell layer highlighted here. And these cells that are contractile are actually sandwiched in between a layer of epicardial cells, which is a cell layer important for the nutrition and maintenance of the heart, and also its regeneration. And a cell layer that is in contact with the blood, which is called the endocardium, and which contains important cells that will participate in the formation of the heart, and in particular in the formation of the valves of the heart. And I will spend some time discussing cardiac valves with you during this talk. And in particular, the valves that are separating the ventricle from the atrium. And these valves are called atrioventricular valves. So they just allow to limit the formation of so-called reversing flows when the heart is beating. So they act as a blocker when the heart is beating, so that you generate a unidirectional flow in the heart as a result of the contraction, which is not simultaneous between the atrium and the ventricle. The zebrafish heart is simpler than human heart. It just contains 2 chambers, but in many ways it's comparable to a human heart. And in terms of developmental processes, there are lots of genetic pathways that are conserved. There are major differences, both anatomically and genetically, and we have to deal with it. We accept that. But overall, it's a very nice system to track and explore both mechanical forces and cell response during the process of cardiogenesis. So the heart in fish takes a couple of days to develop. We agree that after 5 days of development, the heart is almost fully functional. So the valves are there, they are active, and the heart is pumping normally. So a fairly quick process. However, at the scale of the beating of the heart, it's very slow. So the zebrafish heart beats approximately at 3 beats per second. It's something like a 100 micron diameter, so it's pretty much the size of a human hair. So very small. So we need high magnification and specific microscopes to get to that, but overall it's achievable. Now, what we like in particular in the system is that it's trackable, both for looking at mechanical forces and for looking at cell organization. So I'm not sure you see my pointer. Do you see my pointer?
Yes, we could see your pointer.
Okay, great. Thank you very much. So you can see here a set of leaflets. They are not valves yet. They are still growing structure, but you see that, so it's a zoom in this area of the heart. And you see that you have some cells piling up here, and they are in the process of shaping a valve. And you can see these individual cells. So we have a nice resolution that allows us to track both heart contraction, flow forces, all the dynamics associated with the contraction and generation of forces within the heart, as well as cellular organization of the structure we are looking at. So again, a nice system to track both mechanical forces and morphogenesis, even though the timescales are completely different. Here we are looking at seconds, right? Whereas to generate that structure, it will take days. Now, why zebrafish valve? I mean, valves in general are really important. And it's actually a structure that is acting within the heart. And if your valve don't go well, you will be very sick. You can have major problems, and there are families of pathologies affecting cardiac valves, which is still very difficult to treat. But pretty much every structure, for example, in the circulation system, contains valves. So there are valves within the venous system, in the lymphatic system. There are also valves in the digestive tract. It's a universal system that allows the circulation of fluid unidirectionally within the body. And understanding how heart valve develop helps also to understand, or may give some ideas to understand how these other valve structure are forming. And in the lymphatic and venous system, it's also associated with important pathologies. So understanding valve formation is actually extremely important for health. So quickly, I will give you a few elements about how heart valves are developing. You just have to remember that there are a set of genes. That's a complicated gene network that needs to be activated to trigger a very important cellular response at the level of the endocardium, which is an endothelial to mesenchymal transition, so that the cells of the endocardium will invade the space between the endocardium and the myocardium. They will divide, grow, and this will create the basis of a remodeling that will eventually lead to a valve elongation and formation of a valve. Now, there are sets of pathologies called valvulopathies that affect valves specifically. They are very often congenital defects, and actually cardiac valves account for a huge number of these congenital cardiovascular malformation, which are problems happening during embryogenesis. And now we start to associate some gene function with pathologies. So for example, filamin A is an actin-binding protein, tensin 1, Dacus R cell adhesion protein. So these proteins are leading to valvulopathies. However, we don't really know what they do. And the fact that they are associated with the cytoskeleton of the cell made us think that it could be just associated with abnormal process at the cellular level during the program and the generation of the valve. And interestingly, there is nothing much known about the process of valve formation in response to mechanical forces. So there are lots of groundwork to do to tackle this question, trying to integrate mechanical forces with cardiogenesis and endothelial to mesenchymal transition, for example. And a couple of years ago, when I was a postdoc in the US, I was at Caltech in the lab of Scott Fraser. I worked with my colleagues excited by heart imaging, trying to collect this type of images. So a beating heart at 48 hours. We did this movie later on, but all the technologies were initiated at the time, and we were having results that were very similar to that. So starting to have a glimpse of what is the dynamic around heart contraction. And in addition, I found that a gene called KLF2A, which is a transcription factor, was strongly expressed in the area where the valve will form, so this canal. And I could show that heart function was very important to regulate the expression of that gene. And from then, we had an entry point into heart development, gene expression, and mechanical forces, and we pretty much followed that thread. So we followed how KLF2A is regulated, what are the elements that can activate or repress its functions, and this led to a lot of very nice results. Now, I was trying to tell you before, there is not only flow in the heart, and cells experience a number of mechanical stimulation. And if you look at slightly higher resolution, so this is a similar movie, but this time we are focusing on a labeling that highlights the actin. So this transgenic line is called LifeAct EGFP. So LifeAct is a small peptide that binds to the actin, and it's enriched in the area where there is actin. So you can follow the change of the shape of the cells, right, as a result of the accumulation of this label in the cortex of the cell. And you see that cells are enormously deforming, highlighting the complexity of the system at the mechanical level, which is that cells experience shear stress generated by the movement of the red blood cells. But in addition, they also deform and certainly are stretched by the effect of simply the contraction of the myocardium. And this may also constitute an important mechanical stimulus that the cells sense and react to and deal with in order to coordinate morphogenesis. So today I wanted to show you some of the work we are doing in the lab, and I'll try to discuss work on morphogenesis, on mechanotransduction, and on the telomerase transition we discussed in introduction. And this was amazing work made by PhD students, Hélène, and two postdocs, Hajime and René, back then in Strasbourg, and then taken over by people here in London. So let's start by morphogenesis and the process of cardiac valve formation in response to forces. We had to clarify how cells organize in order to make a valve so that we could connect the influence of mechanotransduction and flow with morphogenetic process. And this was actually a very hard endeavor. It was pretty tough because the heart is beating, so it's really hard to follow cell movements in this very active environment. So we had to break down the whole process, which takes something like 5 days, into smaller pieces and develop different types of approaches for each of the stages we would look at. So I will start with the early stage, which corresponds to the formation of the canal, so the atrioventricular canal, where the valve will grow and shape. And it's actually time-wise not very clear. So early on, the heart is a tube-like structure, which is highlighted here. So we are highlighting the endocardial cells. So each of the spots is a nuclei of an endocardial cell. And you can see that as time proceeds, so like in 24 hours, this linear tube is bending in order to get to a nicely shaped heart with an atrium and a ventricle. But you see that there is a strong morphogenetic change that happens here. And we addressed what happens in these cells between that stage and that stage so that the atrioventricular canal forms as this smaller structure. So to do so, basically we tracked cell movement. And we did so through different techniques, and I will not show you all the details. But what we found was that if you look at the canal itself, so if you zoom in here and you just straighten this curved line structure in a line, during the stage I showed you, the area that covers the atrioventricular canal just shrank. So the cells are doing something that allows this canal to shrink, and it's certainly the basis of an important morphogenetic event, because during this time period, the canal forms. So at the cellular level, we didn't know what was happening, and we wanted to address this question. And in particular,
sorry, I have a quick question.
Yeah.
Is there an increase in cell number during this process?
Yes, exactly. So throughout the time of the looping, cells will accumulate. So this is a parameter we had to take into account, and I'll show you that actually proliferation is not affecting at all the process.
Thank you.
So what we did was to try to focus on the cell and not looking at cell shape, but cell volume. And there are different reasons for that. But overall, we thought that that could be a key element in the process. And clearly, people never really looked at cell volume, or I mean, very few, because it's actually very hard to measure. I'll come back to that later. I'll just show you that as time proceeds, as the heart is rotating, there is a major increase of density of the cells within the atrioventricular canal, very clear. So that cells tend to accumulate in this area, which opens the question is whether there is more proliferation there or any change of cell shape there. So what we did was then to measure the volume of the nuclei in each of the fish at these different stages. And we found that as time proceeded, the volume of the nuclei in the canal is actually much smaller than the rest of the heart. So this is the quantification here at 48 hours, where we display the average nuclear volume. And we found that the nuclear volume is much smaller in the atrioventricular canal than the rest of the heart. However, earlier, they are non-significatively different, right? So this is it's a process. Cells will change their volume as morphogenesis grow to get to that structure. Now you have to believe me, we made all the controls, but we could show that nuclear volume scales with cell volume. Cell volume is really hard to measure, especially in such compacted structure. And for just the sake of quantitative analysis, it was much easier to segment and measure the volume of the heart at high throughput in a way. So we did this, but we made all the controls to make sure that this measurement really reflected the cytoplasm and the cell volume per se. Now, what happens in terms of proliferation? Is proliferation important? Well, it turns out, as I told you, the spoiler, cell proliferation doesn't really affect the process. So we could block it, and using a set of drugs that block cell proliferation. And you see that after blocking cell proliferation, as expected, you have much less nuclei, and it's actually really nice. You really store the cell number to the time of the treatment. So we treated embryos at 30 hours and waited until 48 hours. So we really stopped the total cell number just remained the same. But you see that looping is happening. And if you measure the cell volume overall is bigger than the wild type heart, but the ratio are conserved. So there is still smaller nuclei in the atrioventricular canal than the rest of the heart if you stop cell proliferation, suggesting that it's not proliferation, it's not local proliferation that explains this change in nuclear volume, but something that is independent, and we think most likely cell independent, cell autonomous. So we made a couple of experiments to find out then what is the impact of heart function and mechanical forces. And to do this, we use different types of mutants. We have mutants where heart function is completely abolished. These mutants are called silent heart because the heart is silent, kind of beats. And it's just a troponin protein which is mutated, which is an important element in the contractile unit of the cardiomyocytes. And these mutants have a very dramatic abnormal atrioventricular canal. And in these mutants, the nuclei and the cells are much bigger than in the control. So this is the quantification, suggesting that heart function and mechanical forces are important to activate the process of cell volume shrinkage. We then looked at some other type of mutants that affect mechanotransduction, and I will describe them a bit more later on, but these channels are known to modulate cell function in response to mechanical forces. And it turns out that they also have some issues in cell volume reduction, suggesting that these channels are mechanosensitive or sense something in the system that will modulate the nuclear volume and help for the morphogenesis of the canal. So overall, from these experiments, we conclude that the heart function and these mechanosensitive channels are essential for the morphogenesis of the atrioventricular canal. So overall, what happens early on in terms of morphogenesis is that while the tube is bending, cell volume is changing and is associated with movements of cells that I didn't show you, but are there, that coalesce or converge within the atrioventricular canal, so that cell volume change and movement are coordinated in the process of the formation of this canal. So that's for the first step. But what happens later is also very important. And it sounds like a question which is again related to forces and biological function, which is stretch and shear stress affecting the so-called endothelial to mesenchymal transition. So it turns
Sorry,
out
I also just had a question, if I could interrupt for a moment. So these mechanosensitive channels, is it reasonable to assume that basically the mechanism of the change in volume is that these channels are pumping ions? Is that the...
Yeah. So we are actually in trouble here because these channels are mechanosensitive on one hand, but on the other hand, they are also activating flux of water in the cell. So TRPV4 is able to bind to aquaporin, which can be activated by this binding and then lead to the release of water. And most likely this is what happens, which is that for the cell volume to change, cells will have to change ion content and water content in their cytoplasm. So yes, that's a big question now, trying to understand what's the mechanism of cell volume change. I didn't show this to you, but we found that extracellular matrix is important, and in particular hyaluronic acid. And we believe that most likely what happens is that there is an accumulation of hyaluronic acid around the cell, and this creates an osmolarity gradient that the cell has to cope with, or that is important for the process to happen. But if you get rid of this hyaluronic acid, you have the same issue. Cell volume cannot change, and the cells remain big. So we know that they are important. Now the question is, yeah, the mechanism, because you could also argue that maybe cells around them are moving, migrating, and pushing the other, and the cells of the UV canal deal with it by changing their volume. But overall, this needs to be coordinated one way or another. And we see that cardiac function is important. So most likely, it is interconnected in a way. But all right, so for the subsequent stages, there is a much bigger event which is happening, which is this endothelial to mesenchymal transition. And interestingly, there is a long work that has been done a lot in very strong engineering lab, showing that the endothelial to mesenchymal transition is actually very dependent on cyclic strain, shear stress, as well as matrix properties. And actually, it's really interesting. The response of the cells can vary a lot depending on the type of strain, the amplitude of shear stress, or depending on the matrix properties. And sometimes you have opposite effects. So when you have low shear stress, you can activate the endo-MT, while when you have high shear stress, you inhibit endo-MT. So the system doesn't behave very linearly, and is certainly very controlled at the molecular level. Obviously, we had to address this. So one problem is that, again, in terms of morphogenesis, we had to show what's happening, and we found that the origin of this bulge of cell, that is really the premise of the cardiac valve, comes from a strong reorganization of the endocardial sheet as a result of migration of cells. So now I'm zooming on this area, so the upper valve. I'm just showing a piece of the atrioventricular canal again. And we found that cells between the ventricle and the atrioventricular canal are migrating within the cardiac jelly and bringing the cells surrounding them together to create a multi-layer that you can see highlighted a bit better here as time passes. And this is certainly mediated by so-called collective cell migration. But it's not really an endothelial to mesenchymal transition as people describe it in mouse, for example. So there is a long debate and still very open. But most likely what happens is that after this initial collective cell migration, cells have some highlights of endothelial to mesenchymal transition. I won't go into the details because it's an intricated question, and you're certainly not an expert into that, but overall you'd have to first agree with me. But we see that there are some key landmarks of endothelial to mesenchymal transition, even though it's not exactly the same. And one of these landmarks is, for example, downregulation of V-cadherin in the multilayer, as well as activation of a marker of endothelial to mesenchymal transition, which is twist1b or snail1b, two big markers of endothelial to mesenchymal transition that you can see in the area of the heart. So overall, the question is whether our system is nice to track these markers and how they behave as a response to flow. And we are doing so at the moment, but one key result is the following, which is that when we look at mutants that do not have red blood cells, and this has a significant impact on the viscosity of the blood and the mechanical forces in the system, we see some very interesting valve phenotypes, which are much bigger than the control. And our analysis showed that actually there is an intermediate element that needs to be activated by the wall shear stress, which then inhibits markers of endothelial to mesenchymal transition. So what we think is that depending on the stage and depending on the amplitude, most likely, of the forces, cells will react differently and will proceed within the process of morphogenesis, but will activate or inhibit endothelial to mesenchymal transition and many other cellular processes as a result of mechanotransduction. So this leads me to my last part, which is related to mechanotransduction. Obviously, that's a key question, which is that how do cells sense and react to mechanical forces, and what are the pathways activated by mechanical forces within the system? So I will now discuss the work of Hajime. And first of all, I want to make a quick overview on mechanotransduction at the cellular level. You may be expert in it, so I will be very general, but overall, cells have many ways to sense its mechanical environment. It's completely embedded into it. And of course, the overall structure of the cell is mechanosensitive, in particular, the actomyosin network. But the cells also have these linkage protein, right, that bind either to the matrix or bind to the cells themselves. So for the matrix, you have integrin. For the cells dealing with each other, you have cadherin. And we know that these proteins are also mechanosensitive. And finally, there is another set of mechanosensitive proteins that are just mechanosensitive channels. You may have heard of it. For example, piezo-1, after the Nobel Prize being given to Arden Pataputian, the discoverer of piezo-1, amongst other colleagues. But these channels are really interesting because it's pretty straightforward. When you look at cytoskeleton, integrins, or cadherin, you have lots of elements intricated with each other to explain mechanotransduction, whereas with mechanosensitive channels, well, the mechanism is really straightforward. You just have a channel within the cell membrane, and when the cell membrane change shape, this channel opens and there is a flux of ion, so the cells just know what happens, right? So the mechanism of opening of these channels is still very debated for piezo and others. A universal mechanism seems that these channels are connected to the cytoskeleton, but for piezo it may not be necessary, even though there are some indication that it exists. Now, there are lots of mechanosensitive channels. It's a big family. So we spoke about piezo, but we also spoke about TRP channels, for transient receptor potential channels. There are other types of channels specific to prokaryote. And finally, there is TREC1, which is on its own type of mechanosensitive channel. But you see that the structure of this channel varies dramatically. And overall, in my lab, we have studied TRP channels and piezo channels. Once again, because their mechanics, in a way, is much simpler than any other of the mechanosensitive elements of the cells that I described before. So let's have a look at TRP channels. First of all, are they present in the heart? And the answer is yes. So we looked at TRPP2 channel and TRPV4 channel. So this is the distribution of the mRNA using fluorescent in situ hybridization. It turns out that these TRP channels are a bit interdependent. So TRPV4 interact with TRPP2, and TRPV4 is very important to mediate the endothelial cell response to cyclical structure. So these are very meaningful for the vascular biology. But interestingly, so they are both expressed in the heart. So TRPP2 is fairly ubiquitous, as you can see. So this is the heart. We highlight the cell layer, whereas TRPV4 is much more specific to the endocardium itself. So they are present in the right place and the right environment. Importantly, they are cation channels, right? They are not specific, but they let calcium enter in the cell. And well, we have very nice tools in fish to highlight calcium activity in the cells. So we thought that was very important. So this is how it works. We basically express the calcium reporter specifically in the endothelial cells, and this calcium reporter, if you are able to image them fast enough, you can see some activity. Here we are looking at endothelial cells migrating in the trunk. So you see the nice dynamics. But we also looked in the heart, and we could see that there are some calcium enrichment in the canal. So that's the control. And when you stop the function of the heart or the function of TRP channel, this calcium response is abrogated. So we're happy about this, but overall, this led to this picture, right? Which is very linear in a way. So you have mechanical forces. So the transducer would be this mechanosensitive channel, TRP or piezo. Calcium will be activated in the cell, and then something is decoding this calcium, and then you have an output, which would be the activation of KLF2A or other factors that have been identified later on. But really, what interested us was to find out what is this decoder, what is really the transduction, and how is this working, right? How do you read calcium? So this is what Hajime wanted to focus on. And one of the things we were very frustrated with was that all the images of calcium we got so far were very static. And we knew from working in cell culture that when you impose shear stress on endothelial cells, you should see calcium waves, or at least some dynamic, not a static and boring response. So Hajime decided to investigate calcium activity in the endocardium, and for this he tested all the calcium sensor possible existing in the community. And it turned out that amongst all of them, only one gave some very nice results, which is the GCAN 7.9. And the results were very, very striking. So this is a result of one plane imaging of the calcium reporter. So we have inverse LUT. So black means signal and white means no signal. So now looking at the movie, you see we are imaging fairly quickly. We are just looking in one plane, and each time you see a dark spot, it means that there is a cell that generates a calcium flux. Now it's really nice, and you see that it's very specific of the canal, right? But it's moving a lot, so I'll just show you the same movie, but we are showing you just one image for the same cycle of the contraction. So all the movement is avoided. There is still a bit of flickering because the alignment is not perfect, but now as time passes, you can see how the cells are reacting and how much calcium they generate, right? And so you can measure, you can see that there are some pulses or oscillation. I mean, it's not super well synchronized, but sometimes you have those peaks. But what you see also is that cells are just really randomly activating their calcium pulses. They are not synchronized with each other. And finally, you can see that it's very specific of the atrioventricular canal area, and there is no calcium pulses anywhere else. So very local activation of calcium, and we know that this area has the most shear stress generated in the heart. So obviously, we looked at the calcium response in cases where we stop the heart, and fish are doing very well with that. They don't really need their heart early on to breathe because there is enough oxygen in the water. So you can stop the heart and they will swim happily. So not so much collateral damage. And what you can see here is a heart which is arrested, not completely, it's twitching. So obviously its contraction is abnormal. And the calcium response is completely gone. You can wash out the drug. So here we use tricane. And after the washout, the heart is getting back on track. It beats again, and calcium oscillations and pulses are seen again in the canal. So very nice correlation between presence of heart contraction and flow with the presence of the calcium pulses, suggesting that it is mechanosensitive. So to show this more directly, because obviously this is very indirect, we had to go a step further. And to do so, it took us a lot of time, a lot of thinking, and a lot of prayer, because these experiments were our dream experiments. But I have to say, nobody except Hajime agreed to do them. So he was crazy enough to try them, and I really thank him for that, because that gave amazing results. And it comes from a very old experiment, 2003, that were done in the lab where I did my postdoc, where they used mechanical blocking of the heart by simply grafting beads. And what they found was that if you graft bead in the heart, you will stop flow in this heart, because it will clog the heart. Now we decided to use slightly smaller beads, so that the bead would come in, but let the heart beat very well. And we knew that generating an obstacle within the heart would change the flow profile around the bead. And this would create most likely an increase of shear stress. And to really demonstrate this, we teamed with Yap Shun-Wai in our department here at Imperial, who developed some 3D modeling of shear forces of the zebrafish heart. And he just included a bead in its model to show how wall shear stress actually changes as a result of the bead. And he could show that when a bead is present, as the heart contracts, you have an increased shear stress surrounding the bead. So doing the calcium experiments now in the presence of the bead, you can imagine the result, right? So if shear stress activates calcium flexes, you would expect to have ectopic calcium around the bead. And this is exactly what happened. So when you look at now the calcium response in the presence of the bead, you could see that there is a strong activation of calcium, which is never seen normally in the control. In addition, we took use of these beads that are magnetic to directly pull them in a non-beating heart to see what would be the effect of simply the movement of the bead as a result of magnetic stimulation, right? So just focus on this panel for the moment. You will see that we are coming with the magnetic probe, and as a result of moving the magnetic probe, we can move the bead within the heart at will. And this heart is not beating, which is okay again. So overall, there is no activity, no contractile activity. Now, if you look at the calcium response, it's striking. You see that each time the bead is moving, you will generate a response of the cell, which is a calcium response, which is really nice. So we could really safely conclude from these experiments that it's the presence of shear and maybe a bit of stretch as well that activates the calcium response in these endocardial cells and nothing else. So this is the quantification showing that there is a direct correlation between the movement of the bead and the calcium bursts observed in these experiments. So in addition, we could really explore what is downstream of this mechanosensitive calcium activation. And we found that it's none of the transient, none of the stretch channel that I introduced you with. it turned out that it's something completely different, which is associated with an ATP release of the cells as a response to shear stress. And this comes from work that has been done in cell culture by Yamamoto and his colleague, showing that actually ATP binds to a specific set of receptors, purinergic receptors, to activate calcium waves as a response to flow. And we could test this in vivo by injecting or blocking ATP in the embryo, and we could see that blocking ATP generation or digesting ATP had a very strong influence on the number of calcium spikes in the ADC. So you just decrease it when you have not enough ATP, or you activate it when you just release ATP in the heart. And when you play around with the purinergic receptor, valve formation is abnormal. So overall, we also could see that there is downstream targets in the system, which is NFAT, which is an important marker of MT, suggesting that overall, so first of all, this is the NFAT response in the atrioventricular canal. And we could see that in presence of the bead, this NFAT activation was visible. So overall, this led us to, starting from trying to image calcium response, we ended up with a completely different picture of how the cell actually deals with mechanical forces. We thought originally that it's all mediated by TRP channels and a linear activation of transcription factors. It turns out that there is another pathway going through calcium oscillation that activates transcription factor, NFAT, and other target genes to coordinate valve morphogenesis. So overall, all of this obviously is interdependent. So most likely, it's also reflecting different step of the maturation of the valve. Also, you certainly will need some mechanical stimulation to stop the process of endo-MT, etc. This is still a very open question. But overall, I think we have a much better picture of how the cells deal with mechanical forces and in the process of valve formation now. So to conclude, we have a number of ideas. First of all, there is a very important observation in all of these experiments, which is that when you graft this bead and you wait long enough in very few number, because those experiments are really hard, but still in significant number, you can generate a fully grown heart valve, or at least a structure that really looks like a valve, suggesting that mechanical forces per se by themselves can really trigger the whole process of valve formation. It's not a perfect valve, and we call it valvuloid or whatever, but really the influence of mechanical forces is really powerful. And you can imagine that you could use simply mechanical forces and nothing else to try to grow up valves out of sheets of cells. So this is something we are very excited in, and I joined the Department of Bioengineering, so I'm surrounded with amazing people that are very quantitative and very interested in tissue engineering and morphogenesis. And really, we would like to combine this work to try to really get to a better understanding of how we could, in vitro, try to build up tissues in a way that is controlled and where mechanical forces can be controlled or just bypassed, knowing the pathways that are activated. All right, so I will conclude here. Big thanks to all the lab, current and former. Big thanks to Ajime, René, and Hélène. Big thanks to all our collaborators. And thank you for your attention. I'm happy to take questions if we still have a bit of time.
Well, thank you very much for such an interesting talk. So please feel free to unmute yourself and ask Julien any questions if you have.
can I ask a quick question? So calcium signaling shows oscillation. What's the significance of oscillation itself? Because it doesn't look like they're synchronized, or
Yes.
it's very periodic.
Yeah. So the question is whether this oscillatory behavior is important for
Right.
calcium itself,
Mhm.
and what is the origin? So what is controlling this pulsation? So I have no answer for any of those. This is really interesting and really important. So that's something we'd like to explore more. One thing is that this frequency of oscillations or frequency of pulses, because it's not those nice thing periodical, unfortunately. But this may also be interesting. The fact that they don't synchronize and the fact that they are not oscillating at a fixed period is also very intriguing. But overall, people in the calcium field are thinking that there are pathways that are in line or tuned with the frequency of calcium pulses you see. And for example, we saw that NFAT is activated in these cells, and NFAT corresponds to a region of frequency that fits with the current.
Oh, I see.
So yes, there is a possibility that the frequency of pulses actually would activate specific elements within the cell, and you would have a way to discriminate. The cells would have a way to discriminate those frequency peaks, right? Or they may have a preference for that. Now it's hypothetical, I think, and it's really hard to test. But we have some ideas, so that's something we'd like to do. Try to see whether we could hijack the cell with just activating a certain frequency of calcium oscillations, and see.
Yeah, that would be really cool.
Yeah. So we think about optogenetic tools, right? It's not that straightforward, but yeah, this would be really fun.
May I ask something as well?
Yeah.
So you mentioned earlier that it's possible for the valve to fail. So I just wonder, do you have any idea, like, or maybe like what are some of the causes that cause the valve to fail? Is it related to the calcium response?
Yeah. So that's really really important indeed. Now that we have this more complete picture, it would be really nice to have an idea of how the system reacts in the absence of filamin, for example. So filamin is an actin binding protein, so that it may affect pretty much every of the process we described here. Obviously, the actomyosin complex is really important. Now, really what filamin does in this context, it hasn't been described. So I don't know yet, but that's something we should definitely do. And similarly for tensin and Dacus, I mean, all these proteins are cell adhesion proteins, so it could be associated with problem in adhesion and dynamics in the process of morphogenesis. But it could also be anything else. So yeah, we should do that. I agree.
Thank you. There's also a question in the chat as well. I don't know if you see it.
So Gas was asking, what are some examples of the applications of this study to tissue engineering?
Yeah, so that's something we are starting to work on, and it's unclear. But I think one of our idea is to try to see whether simply deformation of the substrate or changing the properties of the substrate would be enough to activate some morphogenesis process without having to do to use chemical elements, you know, trying to reproduce exactly at the molecular level what happens within the cell. So, I mean, right now, it is still very, let's say, immature, and we may not tissue engineer straight away something. We may also just use it as a way to control the mechanical environment and see how we can modulate cell shape, cell response. But eventually, yes, the outcome would be to try to recreate a full heart just by stretching and moving cells. Now, there are lots of examples in which people have shown that the geometry of the matrix organoids or cells are embedded with is really important to dictate cell identity, cell proliferation, and organoid shape. So yes, that's really embryonic work, but that's the long-term goal.
Can I ask one more question?
Yes.
Might be really stupid question, but with all your high resolution imaging data and fluid dynamics, is any of those affected by the fact that the heart is on the left side, the directionality of the heart looping?
Yeah. So, what is the influence of the asymmetry in the whole
Right.
in the whole system? That's a good point. So normally it shouldn't, but we haven't investigated this. I mean, so what we think in general, when you have those situs inversus, there are a lot of mutants where the heart is bending on the other side.
Right.
And we think that it's really a mirror image. And normally people, I mean humans that have those defects are doing okay for most of them. But whether the process is completely normal and symmetrical, I think at this resolution, nobody really has looked at it. I don't know. That's something we should check.
Well, I can't really imagine if everything is mirror imaged and then if the dynamics would be affected. But if it is, it'll be really cool.
Yeah, I mean, when you have those left-right defects, there may be some subtle differences that people haven't monitored.
Yeah.
I mean, so there are lots of defects associated with cardiovascular defect in patients that have left-right defects. But usually they are collateral damage. So for example, you have left-right defects because cilia function is affected, and cilia has a very important role in setting up the left-right axis, but also roles in processes directly in the cardiovascular system. So it's difficult to separate the two.
Right.
But very often people with left-right patterning defect have issues with their cardiovascular system.
Another question. Yeah, so with the simulation that you have earlier, I think by Dr. Chun, with the bead inside of the vessel, do you happen to, I don't know if it's related to the actual talk, but do you happen to know what elastic model that the person used to model this simulation? Right.
I'm not sure. I should I should have invited him at the talk, but obviously all of this is published. I mean, so it comes from a model they designed previously, which is based on 3D imaging, and they really adapted their model to the data sets we had as well. But now I mean, one of the goals is to try to model how the cell shape is changing as the heart is beating, and try to see what kind of repercussion it has on the cell mechanics, and potentially the type of mechanical stimulus it's experiencing. So we are working also with physicists to clarify this. So hopefully I would have more results and more quantitative answer to that later.
Thank you very much. Yeah, it's really interesting. Thank you.
You're welcome.
Do we have any more questions for Julien? Alrighty, I think we'll wrap up here for today. I'd like to thank our speaker Julien again, and everyone for your participation. Have a nice day, and hope to see you all next time. Take care.
Thank you.
Thank you, Julien.